Take two. All right, so folks, I'm really honored that I was asked to be part of this because I have huge respect for the group that put this meeting together, and they've been doing extraordinary work in the region with respect to K-12 education and beyond. And I'm very passionate about water. I see water as a resource. In all its facets, water is a resource, and even contaminated water can be useful. So I just wanted to frame it with what I promised you, is that we think about human activity, the impact of human activity on our waterways, and how we contaminate our waterways on the regular chemical and biological waste, right? And how folks around the world, groups and individuals, are endeavoring to try and ameliorate and reduce this impact that we're having on our waterways. But many SENCER faculty are working on water issues and introducing water into their curriculum. So here's my story and what I've been doing. And it starts in New York. When I was first teaching, I was in City Tech, and I started working on the Gowanus Canal. So if any of you know about the Gowanus Canal or New York's waterways in general, and every time it rains, raw sewage enters the waterways. And some of the statistics are really shocking, like every time it rains kind of thing. Each year, 377 million gallons of diluted sewage enters the Gowanus, right? And that's rain. But when we think about floodwaters, and we know that New York and the region have been inundated with extreme weather events in recent years, floods are also a great source of contamination, and flooding in particular brings with it the risk of public health impacts, right? And here's a story from Texas, from Houston during Hurricane Harvey, where a woman died from flesh-eating bacteria that she had got contaminated with during the flooding. So when we think about toxic sites in your own neighborhood, can be a brilliant teaching resource. So oftentimes you don't even know it's there. It might be right behind you. You don't know it's there. So when I moved to Texas, first thing I did was looked up the Superfund sites. And at first my colleagues were like, oh my gosh, there's one right there. And they didn't even know. So there might be something toxic right in your neighborhood that you can look at. So when you think about the Gowanus Canal, and we think about Superfund sites in New York City, the Gowanus Canal and Newtown Creek are two very toxic sites that have a lot of history. So if you think about these types of sites, the history that they have, the story that comes with these sites, they're effectively living laboratories that you can use to study with your students. Now, it depends what you want to do with your students, obviously, the levels of engagement you want to have with the actual site itself, but you can learn about the story. And if you're a SENCER faculty, you can bring in the social dimensions as well. So if you think about the Gowanus neighborhood, it's basically been pretty run down for a long time, but we're seeing gentrification occur. So we're seeing wealthy folks buying into the region, into the area, despite the fact that there is this toxic waste dump that most people have had to live with all their lives. So it's a huge problem. And sorry, my internet is going to be very patchy, but the murky history of the Gowanus Canal, I am not going to play this video for fear of my internet dying, but I'll share the slides with y'all. And if you watch this Vimeo video, it actually shows a sewage outflow into the canal, and it's absolutely horrifying to watch it because people live right beside it, right? And it happens all the time, So water isn't just nasty though. Water is also beautiful. And so for many of us, and we like to go to parks, and we like to take in little ponds and little rivers and everything else. And so in Brooklyn, there's also Greenwood Cemetery. So if you're looking for a historical site to study with your students, Greenwood is fascinating because it has a huge number of very important people who are actually buried there. And you can go on tours and everything else. They also have a pair of parakeets that nest there that are not native to the region. And so you can go see them. But from my perspective, the water bodies here are freshwater. And so because they're in Brooklyn, they serve as kind of a nice control for me to study. And the neat thing about it is that they have lovely biodiversity, and they vary from pond to pond. So willows and leaf litter. In one, there's a snapping turtle, and we visited him numerous times. And there are slider turtles and frogs and nymphs in the spring. So, it's a beautiful snapshot of biodiversity that's right there in Brooklyn. And alongside that, then the two canals. So if you look at the image, you can just tell that these are two heavily industrialized, heavily urbanized areas in Brooklyn. And these red spots, I just want you to make note of the red spots. There were 4 ponds in Greenwood. There are 4 sites in each of the canals that we're also sampling from upstream to downstream. And these enter obviously into the river. So these sites are considered brackish. They're brackish sites, so salty water at different levels across the gradients of the canal. Now, this is the first chart that's kind of scary, so I'll go slow. So this is relative abundance. Relative abundance meaning compared across, right? So each of the sites. So if you look along here, there are 8 bars, 4 from each of the Superfund sites. So 4 for Gowanus, 4 for Newtown, and then there's 4 for the Greenwood Cemetery, right? And each of the colors indicates a different season from fall to spring. So we did it over the course of an entire year. We went out, we took a sample from each of the sites. So there were 12 sites in total. And the way that we did this was we did it in a single day. So it was a very long day. We basically went out and we sampled every site in succession, brought them back to the lab and processed them. And what we did was we looked for DNA of microbes that were in the water. And what you can see here is these are actinobacteria. Now, actinobacteria, these would be considered to be environmental bacteria. Some of them can be pathogenic, but generally speaking, these are natural environmental bacteria that you'd find in water bodies. And you can see that in terms of abundance, there were more of these types of bacteria in Greenwood Cemetery across all 4 seasons than there were in the Superfund sites, right? So that's neat. But then we think about alpha proteobacteria and gamma proteobacteria. Now, I would hazard to guess maybe some of you know proteobacteria and alpha and gamma proteobacteria. These are bacteria that are associated with poop. So these are fecal contaminants. And you can see that they're very, very heavily overabundant in the 2 sites in the Superfund sites. So this is not surprising. Fall to winter in both sites, we saw an overabundance of these proteobacteria. So this implies fecal matter is getting into these Superfund sites across the seasons reproducibly. So not surprising there. But it's not just our Superfund sites. The rivers in New York City are also at risk, and the Hudson River, just like the Superfund sites, is heavily contaminated with raw sewage. 900,000 gallons of raw sewage enter the Hudson River. So I've been having my students sample the Hudson River as well, and I don't know if we can actually see the petri dish here to the right hand side there. That petri dish is agar that has been supplemented with heavy metal, in this case arsenic, and we were able to pull arsenic resistant bacteria from water samples from the Hudson as part of the course. Now, if you don't want to have your students go out and look at types of bacteria that are found in these sites, you can have them look at data. And Riverkeeper does a fabulous job of having citizen scientists or community scientists go out and take samples and send it into their labs for counts of bacteria that are associated with fecal matter. And you can see the data and you can see it live. So you can have your students do a project on the types of bacteria that are found in the Hudson River as well. So you don't have to get your hands dirty, you can look at data. So second, teaching resource. So when you think about poop and you think about poop getting into the water and the impact it has on human health, the best way to keep poop out of the waterways is to improve our infrastructure. And so I've been teaching my students about toilets for a long time now, and the impact of lacking toilet access can have on communities. So better toilets and better infrastructure. So when I was at the new school, I developed a course, a first year experience that was based upon access of toilets and how toilets are changing the world. Getting very, very windy all of a sudden. And I'm getting very, very cold. But in essence, so what I was doing for this course was project-based learning, having the students work together in groups to design a better toilet. Now, this was just prototypes. They didn't actually go and design a toilet, they prototyped toilets, but also field trips, e-portfolios, and visits from SENCER faculty. Necessarily, or at least leaving the building, we were able to do a lot of this work directly in the classroom. So this meant that students were engaged in high impact practices without us having to go very far. So we're extremely privileged in the new school in the sense that we had a wastewater treatment plant in the building. And you'll be surprised, most universities will have something like this on their campus that you could bring the students to. And I was surprised when I found out that none of the other faculty were doing this. So we went down and we met with the facilities and they brought us around and we learned about poop puree and reverse osmosis systems and everything else. And my students learned importantly about how waste can be used to generate energy in the form of biogas. Now, you'd be surprised to know that the new school actually invents the biogas, so they don't actually use it. But at the same time, the students started thinking, well, that's a problem. Why aren't we keeping it? Why aren't we using it? And I'll talk a little bit more about that later. But also we used e-portfolios. So the students would reflect upon what they were learning about in class. And I have my students use Our World in Data, which has a lot of information on toilet access and availability. And I had them investigate their own states. So what's going on in your own state? Does everybody in your own state have access to a toilet based off the census, right? And here's one of the students commenting on her state, Maryland, and she was very surprised to see that not everyone in Maryland had access to a toilet. So this is an important aha moment for your students when they learn it's not just faraway places, but right here in the United States. And because we're SENCER faculty, we can also leverage the fact that we have fantastic colleagues. So Cheryl Broverman is one of my heroes, and she established a school in Kenya with her Wiser Foundation, and that's changing access for girls to get access to education, but also to get access to safe sanitation and toilet access as well, so they can stay in school. And it's changing lives in Kenya. So that's another awesome thing to be able to do as a SENCER faculty. And then lastly but not least, this is one of the prototypes. So here's my students. And the fabulous thing about this was that I set them the challenge of making a porta potty, a new porta potty that had to be sustainable. And they worked together during the pandemic. So this was awesome. They were in different countries, some of them, and they still managed to put together a cardboard prototype of a toilet to kind of describe what was going on. And the thing I loved about this is a composting toilet. So they built a prototype porta potty that incorporated a composting toilet. That's a water-free toilet for those of you who don't know. So more sustainable, more resource savvy than these water using toilets that we are so used to. So teaching resource number 3, biogas. You would have heard me mention biogas. So when I talk about biogas and I explain to students that they can make gas from poop, this is usually a huge moment for them because they're like, wait, we can make money? So we can actually use this to make money. And yes, you can. So many groups around the world are looking at this as a way to kind of make the economy, circularize the economy, and trying to figure out the limitations of biogas and how and why it may not work. It's been used for hundreds of years by many groups around the world, where they're actually capturing waste and using the gas for cleaner, effectively for cleaner cook stoves, and preventing people from having to go out and collect wood for fires and everything else. So it's a hugely important industry, and I'm hopeful that it gets even bigger. But what's cool is that if you go to this website, the Water Environment Federation, and you go down to anaerobic digestion, you can actually see where these kinds of facilities are actually being used, who's using biogas around the country. And if you look here, if you remember, I said Newtown Creek is the Superfund site. There is a wastewater plant in Newtown Creek, huge big wastewater plant in Newtown Creek, that is in fact using the biogas that it generates to, guess what, to heat the digesters. So the digesters are actually making the gas that is being used to heat the digesters, which is very cool. And we're seeing this being used increasingly across the country. So this is another area that you can have your students investigate with respect to implementation of biogas. Who's using biogas? What is the energy output and everything else? So that's something you can think about as well for projects. Okay, so teaching resource number 4. So waste and poop and all of that ties very closely into public health and public health surveillance. And so I've been doing, as part of the COVID pandemic response, I've been working with New York City to look at wastewater surveillance as a new technology for COVID surveillance. And so for this, I'm going to slow down a little bit and just walk you through it. So effectively, I'm sure you've all heard about COVID being detected in wastewater, and it's being detected across the country. In many cases, it's being used as a kind of an early warning system to say that we're going to see increased numbers of cases. But we actually were one of the first groups to do this in the United States, whereby we basically take wastewater from the wastewater plants, we isolate the virus, we take the RNA of the virus, we convert that into DNA using reverse transcriptase, which is an enzyme from a virus. And then we basically do RT-PCR to identify the presence of the virus. And then we do different types of sequencing to see what's happening with respect to the variants. So you've been hearing about nothing but Omicron for the last few weeks, but that's how we do it. We actually sequence the virus from the wastewater. And you might be saying to yourself, well, why do that? Because part of the reason why we want to do this type of surveillance is because viruses can evolve over time, as you know, and this can have a huge impact on the therapies that we're using. So the first thing people were saying when Omicron emerged was, will the vaccines work? Will the therapies work, and so on and so forth. And we had some kind of information that said that perhaps maybe not there may be some changes in the virus that allow it to escape therapies, that allow it to escape vaccines, and that could have huge implications for what we're doing to respond to the pandemic. In addition, we're relying on this clinical testing. And I know in New York City, I've seen the news report, 6 hours to get a test, right? But the thing about that is that the throughput for this is very, very problematic. You can only, I see Catiune's liking the infographic. Catiune, this is all graphics from a New School graduate, Molly Metz. As you know, she is a graphic designer extraordinaire, and she's available and willing to do any work for y'all. If you want to get some beautiful graphics, it's all Molly Metz. Though the thing about these samples is they're very expensive. So imagine you're getting one sample from a human, whereas if you're doing a wastewater sample, you're getting hundreds of thousands of samples from the same RNA prep. So it's also biased because a lot of people only get tested if they've got symptoms or they suspect that they've been exposed. It's biased by access. You are many places around the world you can't get tested, and in New York and other places around the United States, we just can't get access to the tests, right? So this is an issue. So it's biased by geography and it's biased by access. But the difference between wastewater and all of this is that the virus actually lasts in wastewater for quite some time. So we don't have to necessarily worry about the signal going away. We can actually get entire households to entire buildings to entire regions of the country by doing wastewater surveillance. So in essence, we just get one composite sample of water, and then we can isolate all the viruses that are in the wastewater sample, thousands of individuals from one sample, and it's much more cost effective, right? And the neat thing about this as well is that it's safe. The virus isn't viable, so that so far at least we've not detected this. Do you need an IRB protocol to test? Not an IRB protocol, but you need an IBC protocol. Fernando, you'll need to get IBC approval, but I can help you with all of that if you want to do it. No problem. Campus surveillance would be cool. All right. Okay, so what were we doing in New York? 14 plants, all 14 plants. So we reached out to the DEP in New York City and we asked them, we basically volunteered our services and say, hey, like, we'd love to help you. And it was myself, John Dennehy, and Monica Trujillo from Queensborough Community College. We put a team together and we offered our services to the Department of Environment Protection. So 14 wastewater plants, 8.5 million people covered by that surveillance, 1.2 billion gallons every day. And we're able to identify all the circulating variants in New York City using this technology. So the RT-PCR was kind of cool. This was the first thing we had to develop with the DEP. They, in fact, had no protocol for this. So imagine that. Queensborough Community College and Queens College and the new school, a little trio of schools, helped the New York City Department of Environmental Protection to do this. How quantitative is it? Is it presence or absence? It's very quantitative. We can get it down to copy number levels, Roberta. So we have standards that we use, and we're actually in the process of getting absolute quantification using digital PCR right now, but we can actually standardize it and get it down to copy numbers per person and so on and so forth. Excellent. Yeah, I know. It's really exciting. So we take a composite sample, and this will be familiar to many of the faculty here. It's very, very similar to what we've been doing before. So for many of us, this will be second nature. If you're isolating viruses from water or you're isolating DNA from water, it's very similar. The only difference is that we use Trizol, which is really cheap. We use Trizol to isolate the virus from the water. There are a few additional steps. We have to pasteurize the water. So that's the 60 degrees for 60 minutes. And then we do the real-time PCR. And so we've been doing it for over a year now, almost a year and a half, in conjunction with the city. And this protocol was published. So very exciting. We were one of the first published protocol in +1 collaborative effort. And on most of these, by the way, are undergrads, graduate students, and master's students from Queen's College. So sequencing. So my role in the project was sequencing, to help with the sequencing. And what I did was I used what we already knew. So for many of you, you know that I've been doing microbiome work. So what I said to myself was, if we're doing the microbiome, wouldn't it be the same thing if we were to do targeted sequencing for the virus? And what we did was we targeted the spike, which is what you're mostly seeing variants occurring in the spike. The spike is the part of the virus that's actually used to bind to the receptor of the host. So in essence, if the variants are in the spike, we can detect them with targeted sequencing, just targeting that little piece of the virus. But we're also trying to do whole genome sequencing as well. And I'm extraordinarily privileged because I have a sequencer. And this is a benchtop sequencer, which is not extraordinarily expensive. It's about $20,000 to $25,000, and the reagents are pretty inexpensive as sequencing platforms go. So it is something that you could potentially use with some school money, to get a sequencing program up and running in your school. But this is what it looks like. So we're actually doing two different types of sequencing. And we have some collaborators that are helping us with this. So with the iSeq, I could get about 333 base pairs of the receptor binding domain of the spike. So if you look up the top here, this is the virus. These are the different genes of the virus. It's really small. It's only about 30,000 base pairs long. It's a tiny little virus. And then we're looking at a region of the spike, not the whole spike because we can't, it's a tiny little piece of it, but it gives us enough information to detect important mutations. And these are some of the mutations up top that we were able to identify in the spike using this method. And this is very complicated, but because we're doing deep sequencing, I thought I'd just mention this to you. So most of you are used to Sanger sequencing, where you sequence just one sequence for your DNA samples or whatever. The thing about wastewater surveillance and NextGen is that when you're sequencing, we use deep sequencing. And so this is where you get multiple strands of the virus. Basically, you get multiple reads that correspond to the region of interest, right? Now, the challenge with wastewater is that we're getting all the mutations from all the people. So it's a mixture. It's a huge mixture of variants. So we basically have to pull out the mutations and look for combinations of mutations to infer what variants are present in the sample. I see another question in the chat. Okay. So this is what we were doing. So we basically took the data from New York City, from people, from the samples from people, and these are the pie charts on the left-hand side. And this is for February, March, and April of 2021, last year. And we compared it to the wastewater. And what you can see is it's not completely identical, but it's very similar in terms of the colors, the distributions of the variants that we saw. And what I want to point out to you, though, is that something that was very significant was that when you look at these pie charts, can you see this dark blue color here? This dark blue color here corresponds to variants that we didn't see in people. So these were groups of mutations that we were calling unusual or novel or unknown variants that we couldn't see in people, because all the other variants were being represented in the samples from people. So this was a hugely important finding, because at the time we had no idea where these weird mutations, combinations of mutations might be coming from. And when we looked at it, what we were able to see is over time, and this is really important for surveillance, disease surveillance, you have to see the same groups of mutations over time. So what we saw was that when we looked at it in different plants, wastewater plant 1, 2, 3, and 4, each of these represents a group of mutations. They looked very similar over time. So remember, these are composites, so this is a mixture. Because we're doing these small pieces of DNA, we can actually infer that these are physically linked to one another. So that's the difference. By doing these small reads, they're physically linked because they're on the same amplicons, the same pieces of RNA. So we know that these are physically attached to one another. But if you look down the bottom, this one was really frightening, because you can see there's tons of different mutations here, tons and tons and tons of them. So we were really worried about this. And so we wrote it up, and it's actually been accepted. It's in Nature Communications, and it's going to press shortly. But this is our preprint, and you're all welcome and encouraged to go there and have a look and tell us what you think. But we're also doing whole genome sequencing, because whole genome sequencing tells us what's happening beyond the spike, right? So because we're not just interested in the spike, there are things that could be happening in the rest of the virus. And for that, again, we're doing targeted sequencing, or tiled sequencing, I should say, across the entirety of the genome. It's way more complicated. So instead of doing just one PCR, we're doing hundreds of PCRs across the entirety of the genome. But the thing about this is you can see the variants that are occurring in regions outside of the spike. And by doing both, we can basically say, well, week to week, we see these variants occurring in the spike. And then week to week, we see these other variants occurring elsewhere in the virus. And that's really important, because we need to be able to do both. We need to be able to see what are the variants that we know, and then what are the variants that we don't know that may be emerging, that may be new variants that are coming. So if you're interested in who's doing this, this is another thing that you could do with your students. So you don't have to do the sequencing or anything like that. You could do research on who's doing the sequencing and who has access to the technology. So if you look at this database, this is COVID Poops. This was a project that was funded by NSF. They're actually monitoring who's doing this type of surveillance. And I will give a prize to any of you who can tell me, where is the most sequencing being done? It's obviously being done in the global north. The global south is lagging behind in terms of sequencing. So access to resources and technology is still a problem in the global south. It's not because they don't have access to the expertise. They have fabulous scientists. They just don't have access to the equipment and the reagents and everything else. It's very expensive. So we're working on that with SENCER and some donations that we've received. So I'll keep you posted. But this you should check out with your students. They'd be intrigued to know who's doing it and how. So as I said, the National Centre has received funding from a donor that is going to be supporting some fellows, water fellows that are interested in wastewater surveillance. And we're specifically recruiting women of color who are doing this kind of work to help us develop educational materials. We're also funded to help support technology developments in the global south And so we're working that, and this work is being done with Monica Trujillo from Queensborough Community College again. I just want to give her a plug for this. And the reason why we got this funding is through, we appeared on This Week in Virology, and we basically put out an appeal for funding. And so we were very lucky to get this funding. We're very grateful to our anonymous donor. So you might be thinking to yourself, well, what does this mean? The research and development in wastewater is huge. So for in terms of students and projects, you can have your students do microbiome analysis. They could be looking for antibiotic resistance genes. They could be looking for different viruses. They could be looking for parasites. There's tons of things we could be doing. And this type of technology allows us to respond quickly. So if you have a scheme whereby you're doing rapid surveillance, you can say, well, it looks like we're seeing an upsurge in this particular pathogen. So we should start looking for it in the people, not just the poop. But also you can consider course-based research experiences. You can integrate this into your courses and have students do different aspects of it. They could be doing the data analysis, they could be doing modeling, they could be doing all sorts of things. And they could also be looking to do research experiences at the sites themselves. So the DEP, the DOH, they have internships that will be looking for folks who are interested in this kind of work in the future. And we see this as a huge opportunity to increase the workforce in STEM, because when we look at who's actually employed in terms of molecular biologists at the DEP, there's not often a lot of folks from our communities, the students that we're trying to teach and reach in our minority serving institutions and Hispanic serving institutions. We need to diversify the workforce in water technology, and this is across the nation. So what can we take from this? Everybody poops. It's a natural resource. It's unlimited. We can use it for stuff, right? So we can use it for gas. We can use it for surveillance. We can also use it to monitor infrastructure and the impacts of climate change on our water bodies. So wastewater is hugely important for that. So you know if there's a leak in the sewer, if you see poop where you shouldn't see it, okay? And there are many ways we can do it. So it helps us reach the SENCER ideals. If we teach through water and water-related issues, we can teach students about the power and limits of science and what science can do for society. It's very important. And lastly, you can also consider the fact that access to this tech is very limited, right? So we need to find ways to increase access to the technology and leverage what we've learned so that we're ready for future pandemics. So that's the last thing I'm going to say about that. So I'm just going to chat real briefly. Oh, you guys are lovely. Okay, on we go. So the last thing I just want to do is give a plug to SENCER. So I am not the only person who's doing this kind of work. There are tons of faculty across the SENCER community who are doing this work and have been doing it for much longer than I have, right? So one of the things we have to remember is that we are a community, and our resource, the greatest resource SENCER has, is all of you, right? So we're doing all sorts of great things. So I'm going to give some plugs, some recent events. So this event from Hawaii, the group, our SENCER state, Hawaii state, they did a fabulous Grand Challenges of Water conference, which was all about collaboration through water, Indigenous knowledge and sustainability. It was a fabulous conference, highly attended, and lots of great collaborations came from it. But they're not the only ones. We've had Montgomery College and the SCI New England meetings. They've had meetings on water. So I think there's a huge interest among the community to build a community around water issues and issues with respect to access and health, public health with respect to water. So we're not alone. We all are interested in this, but we also have loads of examples of curriculum as well. So we have first year experiences, ones on the Chesapeake Bay, others on the Chicago River, where we're looking at project-based learning. These are all available, and I have the links for you at the end of this. We also have projects on the Flint water crisis, and I know Bhavani is an expert in this, but we have some examples from chemistry where they're looking at this, and also environmental geology as well. And the last thing I want to give a plug to is, as you know, your group is epic with respect to K through 12. We have some models of river courses that are tailored for pre-service teachers as well. And lastly, but not least, non-majors courses that are focused on water as well, science on the Connecticut River, and also then the power of water from Alex Fink. So check out what we have on offer. I have some of the links here, and I'm going to be building up this resource as we go. So anything that you have to share, please do, and we'll put it up and share with the community. And that's it for me. I'm going to give a shout out to all my collaborators. I couldn't do this work without all the fellows and students and postdocs and collaborators that I have, the various institutions that I've worked at, funding from NSF, funding from QIAGEN, support from QIAGEN, and lastly to all my colleagues at SENCER, who I couldn't do this work without. None of this would have happened without my introduction to SENCER. So I'm grateful to all of you, and I'll stop sharing there and open the floor to questions.
Wow,